would like to welcome you to a recorded science session focused on the DecodeME project. My name is Chris Ponting. I'm a researcher, very much interested in ME and finding out what is the genetic, what are the genetic causes of ME. Just again want to welcome you to this meeting and to set off with an apology that we have reached our capacity of 500 people for the event on Zoom. And that's why we set up the opportunity for people to join via Facebook. And we'll fix this next webinar, and we'll make sure that we have the opportunity for more people to join us via Zoom. So again, I'll introduce myself, and then I'll introduce Shona, who's kindly joined us today. My name is Chris Ponting. I'm a human geneticist, and so I'm interested in why people are different from one another in a whole variety of different settings. And in this setting, obviously, what I'm interested in is finding out which are the letters from among 3 billion in your DNA letters change the likelihood that any individual is going to be diagnosed with ME. So that's me, and I'd like to now pass over to Shona, who is also part of the DecodeME team.
Hi, yeah, it's a great privilege to be able to join this webinar. My name's Shona Kerr. Like Chris, I'm also a human genetics researcher up at the University of Edinburgh. However, the whole field of ME/CFS is relatively new to me. I'm learning all the time. My background is in what we call population genetics, human population genetics. So not specifically looking at people with a disease or condition, but trying to study the whole population. And when Chris began formulating the idea of what turned into DecodeME, we started chatting about it and realised that although there are many differences when considering a cohort of patients, there are also lots of similarities, particularly at the practical level. So I'd say that's really my kind of role in the project, is to try and advise and bring my experience of being involved in setting up other cohorts. And yeah, just to sort of finish, I'd like to say just how interesting it is to be working closely with people affected by the condition in this co-production that's really quite an unusual construct, and I think a really useful and important way forward for human genetics research.
Thank you, Shona, and it's a delight to have you as part of the team. So what we're going to do now is I'm just going to talk you through some of the science behind genetics and about the genetics of ME. And then we're going to go into a Q&A session. We'll be fed some questions live, and also we have some questions that people have sent in ahead of time. I apologize ahead of time. There might be some background noise in the house. There are people who are working nearby. I apologize if that happens. So what I'm going to do is share my screen, and hopefully now you'll see some slides and see that this is a project that is run for us out of the MRC Human Genetics Unit. It's funded by the Medical Research Council and the National Institute of Health Research. The important thing is to say at the beginning is that we know that ME/CFS is in part genetic. And that's because of previous research, such as this one done over 10 years ago, showing that the relative risk of someone being diagnosed here with CFS is increased, and this is in the red box, if they have a first, second, or third degree relative. So you have about a twofold greater likelihood of being diagnosed if you have a first, second, or third degree relative who's also been diagnosed with CFS. And there is other information around which points in the same direction. So what do I mean, and I said this earlier, what do I mean by the fact that we have letters that are different in our genomes? So what I'm showing here is 2 copies of a genome. And you have 2 copies because you gain one from your mother and one from your father. And so if you compare those 2 copies side by side, you will see that essentially they will be identical, except in about 1 in 1,000 places where there will be a difference. And this difference we're going to call a SNP, which stands for single nucleotide polymorphism, but I'm also going to call it a letter change. And here the letter has changed from a C or a G to an A or a T. And I'm saying C or G because they are paired in a double helix. Anyway, the important thing is that there are these changes, and people have looked at these, changes and have asked whether the changes predict whether someone is more or less likely to be diagnosed with ME or CFS or ME-CFS. So here's one such study which has said that the letters in a gene called the glucocorticoid receptor gene, and has said that there are letters that predict whether someone has chronic fatigue syndrome. And that such study has been done, a whole variety of different genes and different parts of the genome. And what we have more recently done, and we've published this in a review with Joshua Dibble and Simon McGrath, is to say, let's look at those predictions and ask whether in a very large data set in the UK Biobank, whether we are able to see those predictions repeated. And unfortunately, no, they're not in general repeated. If they were, then these bars on the left here in blue would be very high, and these bars on the right here would be very low. That is not the case. So unfortunately, we don't see any replication of gene by gene comparisons of letters. So instead, we look at the whole genome. We look at the genome-wide associations. And this is what we're wanting to do in DecodeME, is to have a genome-wide approach, a genome-wide association study, a GWAS. And what this does is look at the DNA in chromosomes. And these are the sock-like structures at the bottom here. And at every point in which there are changes that are common in people, we ask whether those common changes are more frequent among people with ME versus others who don't have ME. So if this is so, that there are more people with this letter in cases, people with ME, than in controls, and that this is statistically significantly so, then they predict whether someone is a case, someone with ME or a control. What's important to say at this point is that these letter changes do not categorize people. They don't 100% separate between people with ME and controls. So here's a new article that's just come out by colleagues in Norway, who have looked at people with ME, ME-CFS in 3 different cohorts. Their Norwegian cohort, a Danish replication cohort, and a data set from the UK Biobank, which is what I was just referring to. It's a very interesting study and a very well done study. What they conclude, and I'm just cutting to the chase here, they say that we did not find any ME-CFS risk loci displaying genome-wide significance. And pictorially, this is what they see. They see these dots across the x-axis here, where all of these are different chromosomes. Each of the dots is a place where these letters commonly are different among our genomes. And if we were to have one of these dots higher than this red line, higher than this point here, then that would indicate significance at the genome-wide level. There are none. There are no such points. So they said we did not find any associations at the genome-wide significance level. They did say, just trying to get rid of this box at the top. Sorry, I haven't been able to do that. That they did indicate that there might be some association to a particular gene called TPPP. And they said that there was some modest association. But when you look at the large data set for CFS in UK Biobank, again, there are no such dots that go above this red line. So no strong evidence for this gene. What they then say, and I appreciate what they've said here, that DecodeME will enable, as it is a larger and more powerful cohort, will enable a more definitive investigation of associations between genetic risk factors of ME. So that points to our study. So our study is questionnaires from home. We send a saliva collection kit to your home, and we ask you to send that back via the regular post. And we do all of the DNA processing and the data analysis away from your home. So we ask you to do questionnaires either online, which is our preferred option, or if that presents difficulties, then via a paper questionnaire. And we ask you to do that if you are expected to pass criteria. That means that you've had a professional diagnosis of ME or CFS or ME-CFS. You're 16 years of age or older, and you've got a diagnosis of any type of ME or CFS, ME-CFS, mild, moderate, or severe. We're wanting to recruit to the study 25,000 people, 20,000 people who were diagnosed because of an onset prior to COVID, the COVID pandemic, and 5,000 who specifically have been diagnosed with ME-CFS because of their COVID-19 virus infection. And that is a newer part of our studies since last year. So we are going to, for those people who pass the criteria, and if you don't pass the criteria, it doesn't mean to say you don't have ME-CFS. It does mean that we just have particular criteria that have been set for us internationally and by our funders. And so we will ask for those people who do pass those criteria to give a saliva sample in a tube that goes back into an envelope that goes back into the post and is sent to the Biosample Center in Milton Keynes, who will extract the DNA. And from there, the DNA goes over to California, and it is analyzed there in exactly the same way as was done for the UK Biobank, and they send us back the data. All of this is done in an anonymized way, and we have strict confidentiality agreements with all of our partners, which have been thoroughly vetted by our institution and by the ethics committee. So then we do the DNA data analysis. We ask this question of every part of the genome, whether there is a letter that is substantially and significantly more frequent among people with ME than in others. We're able to use the UK Biobank in general for our controls. So that's it. We will hopefully be able to find these dots that lie above the red line, which are indicated here in these red circles. And what does that show us? That shows us that there are genetic risk factors for being diagnosed with ME. But that's not where we stop. We will go as fast as possible to understanding the genetic contributions to ME as part of what we already know about biology through disease networks, how molecules work together. We'll use every type of approach that we can, high-powered statistics, artificial intelligence, machine learning. We'll fold in drug databases. We'll ask whether existing drugs are likely to reverse the symptoms observed because they are targets of the genes that have been pinpointed by our genetic study. And then it's not up to us, but up to pharmaceutical companies to take those target genes that we've been identifying and to take them forward for trials, which take, unfortunately, very many years and involve many thousands of participants and can take, as it says at the bottom right here, several years to complete. But the reason that we start with genetics is that this whole process of going to trials has a very high dropout rate. So the number of targets that go forward drops very substantially, very quickly. But if you have genetic evidence behind those targets, then that reduction of targets over time is very much slowed. So there are so many people I have to thank. I just want to do that here on this slide. Lots of people working very, very hard indeed. So thank you very much to all of them. And please do pass on to anyone our DecodeME website and do get involved. So I'm just going to stop sharing now. So hopefully I've been helpful to you to understand a little better of what the DecodeME project and why we've been looking at genetics. So what we're going to do now is look at some of the questions that have been posed to us already via email, and I'll start with one that was sent in earlier. And I have to say at the start that I'm not a clinician, so I won't be able to answer any questions relating to clinical aspects of ME/CFS. So I'll take one which has just come in, which is, can you explain a bit more about what is meant by genome-wide association? So I'm going to put Shona on the spot with this, because I don't think I've done a good enough job on that in my presentation. So perhaps she can give it a better go than I did.
I can have a go. It is quite complicated and there's a lot of terminology to learn as well. But the way I understand it is, so the genome is every bit of our genetic material. There's no prejudgment beforehand. It's everything is thrown into the experiment. And we're looking for, as Chris explained, a difference between one group of people and another group of people. Here, cases of ME/CFS and controls that are otherwise closely matched to those people, but don't have this condition. And we're looking for these letter differences that Chris mentioned. The problem is we all differ in lots and lots and lots of letters in the genome. That's what makes us us. So we have to work out which differences in these letters are statistically significant and between the two groups. And there's a lot of clever ways to do that, but the statistics eventually pan out that unless the probability of this happening is by chance is really, really low, then we can't say it's genome-wide significant. And that's what happened in the Norwegian paper. Solid paper, interesting, really great preparation for DecodeME. But what the statisticians would say is underpowered, and that simply means it looks like there weren't enough people in the study to give a genome-wide significant result. By designing DecodeME to have many more cases and good numbers of controls, we hope that we will start to uncover these genome-wide significant signals.
That was so much better than I did. So thank you very much, Shona. So there have been questions that have come in about whether we are looking across the whole genome, and the answer is yes. So we are looking at mitochondrial DNA as well. And then there's also a question about whether we are looking at not just letter changes, but word and paragraph and chapter changes, larger genetic deletions, micro duplications. The easiest answer to say is no, we're not, except when the letter change near to it actually is a good predictor of those. It tags it. So it's a complicated answer, but we won't be able to look at everything in this part of the study. But we have kept to one side, or we will keep to one side, half of everyone's DNA sample, so that when there is enough money, we will do the whole genome sequencing that will enable us and others to find the more complex changes to the genome. So there's a question asking whether people with long COVID or post-infectious Lyme disease can take part. So the answer is, if you've got an ME diagnosis, then please do take the questionnaire, see whether the criteria fit with you. If you've got a diagnosis of long COVID or post-infectious Lyme disease and not ME, then you won't be considered to be a good participant for this study, but of course you will be for other studies. So there are long COVID genetic studies that are ongoing, and we are talking with those people running those studies actively. So, fantastic question asking, why does ME affect more women than men? So we don't know the answer to that. It is true not just of ME/CFS, but diseases that are autoimmune, which makes us ask the question, is ME/CFS an autoimmune disease? And there is active research ongoing as to why there does seem to be a female bias for autoimmune diseases, but we actually don't know the answer to this question. So there is a question about whether we'll be able to identify risk factors that are in common between ME, ME/CFS, and other diseases such as multiple sclerosis. And the answer is that when we get all of the data and we see what are the genetic signals for ME, then we'll be able to compare them with any disease. And we will compare them with all the diseases for which we have the data. And then in the comparison, we'll say, are the genetic signals overlapping with the genetic signals in multiple sclerosis and other diseases, or are they not overlapping? And that is part of our analysis plan. There's a question about how, maybe Shona, you can handle this. There's a question about the UK Biobank will have people who pass the selection criteria or might do so. Is this something you're happy taking?
Sure, yes. I mean, I think the UK Biobank is a remarkable resource. 500,000 people volunteered to give up really a lot of personal and health data and donate their DNA and sometimes other samples. and many research projects across the world are benefiting from that resource. And DecodeME is one of them. We have the permissions in place to use so-called control people from UK Biobank. So we've got 500,000 to choose from, so we can be quite choosy and try to get the absolutely best match set of people to our cases. And that in itself is actually quite a big project, quite a complex piece of work. So we won't include people who report ME/CFS, and there's a couple of other exclusions, and we'll try and do things like age and gender match. So I think, without sounding like I'm passing the buck, it really is just a complex process to get the best match. I think the questioner is perhaps when talk about the 16% in controls, I think, Chris, you had that on one of your slides. You know, I think that was a hypothetical difference between cases and controls. It's actually, if you read some of these GWAS papers, and I have to say they're often very densely written, the differences between cases and controls in the frequency of these key letters in genes is actually sometimes really quite small, but because everything's done so carefully and the cases and the controls are so well matched, this can still be meaningful and statistically significant. But it's very much pointing to a signal which then further work really unpicks. But one of the reasons I'm such a fan of genetics is that it is hypothesis free. Every gene in the body is being surveyed, and I think it's true for all sorts of complex conditions that all the predictions just got blown away once the GWAS results started coming through, and really whole new avenues of research and eventually treatment do get opened up as a result of this amazing GWAS technique.
Thank you, Shona. So there's a question about the criteria used, and the questioner says, presumably not FACUDA. No, not FACUDA. We're not using FACUDA. We're using the Canadian consensus criteria and the IOM NAM, those two criteria. You would have to pass the Canadian or the IOM or both, and some other criteria that we put in place. So there's a question as to whether DecodeME is expected to have as an outcome a diagnostic test. So no, we don't expect it to generate a diagnostic test. What I expect it to do, however, is to improve diagnosis. And by that, I mean that people will be believed more when we hopefully show in this study or in subsequent studies that there are definite genetic risk factors for ME. And that will help to overcome what people quite rightly consider to be that people are stigmatizing individuals with PMA, with ME, and we'll be able to overcome that stigma with the scientific evidence for genetic risk factors for ME. And that will help the diagnostic rate and overcome what is this appalling delay of median 8 years between initial symptoms and diagnosis. So they're just looking to see what other questions that there are. There's a question. Yes, Shona.
So I'm just going to say, I think a few questions come in asking, will we be collaborating with this researcher and that researcher? And I think that's, it's really interesting to see how participants are so engaged with the scientific literature and are reading papers and, wanting to know that we're all working together. And I think that is a really important point for DecodeME is our transparency and our plans to be as open as possible. So yeah, I think maybe there's a few questions we could answer there. One of the slightly complicating factors is that we do have to be very careful about sharing data because it is private personal data. And I think Chris already mentioned the number of agreements and contracts and safeguards we've put in place for all data handling and de-identifying samples, for example. But when it comes to actually reporting results, our aim is always to make these in the public domain. And in fact, GWAS is arguably one of the most collaborative fields of scientific research, certainly of biomedical research. And it's partly because from the early days it became clear that to get this power, groups did need to work together. And one of the quite complicated things to explain is that we don't need to necessarily share individual data. We can do analysis, generate so-called summary statistics, and then these can be added together in a de-identified way. There's no personal information there, and that can allow other findings to be made. So DecodeME is going to be an absolute step change in the numbers, but it will by no means be the end. There will be other projects after it that will just clarify and expand on the current work.
Thank you, Shona. There's a question about the pre and post-COVID, whether it refers to the onset of symptoms or to formal diagnosis. It's the onset of symptoms. So if someone had ME symptoms prior to the pandemic and was diagnosed after the pandemic, then they would be part of the pre-COVID set, the 20,000. Question, does ME/CFS have a neurological basis? That's why we're doing this study. We'll find out. The question that came in before we started, do we have a rough timeline when results might be published? So this year is the data year. We're collecting the data, the samples and the data. Next year will be the analysis year. And the final year will be, in 2024, will be the dissemination year. That's the rough timeline. Then there have been questions about timing, about, have all the participants been selected yet? No, we have only started with a few people to stress test our systems. And boy, are we glad we've done that, because it has become clearer to us that we're not doing a good enough job with the questionnaire. And so we're doing a root and branch revision of that. The latest thing that we're going to do this week is to pass it back to see whether that requires a thorough ethics review or not. But the whole reason why we're doing this is for people with ME. And so we have to do as good a job as possible. And so we really do appreciate people's patience that we're not yet going out to everyone who would wish to participate. But the initial participation has uncovered so many things that are going to substantially improve this study and give a much greater likelihood of success. And that's such an important message for us to give you today. So there's been a question before we came online as to whether we're going to group patients, or people with ME. So we have 25,000 people. Are we going to be able to use questionnaire data to say, oh, well, this person is part of a type 1 or type 3 or whatever, ME/CFS. So we are going to do that analysis and find out whether we can basically say whether people are in one type or another, and then compare the DNA of the people in the different types to see whether the different types have a different DNA basis to them. How confident are we that there is a genetic signal for ME/CFS? Well, there is this likelihood, this greater chance that someone has a diagnosis of ME/CFS if there is a close relative with ME/CFS. And that's what's giving us some confidence that there is a genetic signal. The other thing to say is that there have been so many different conditions over the years that have been thought of as not having any genetic basis whatsoever, but in recent years have been found to have an abundance of genetic evidence for contributions made. I don't know if you've got an example of that, Shona.
Yeah, I think it is a really important question because we are biological creatures. Of course, nurture is important, but nature really does underpin so much of our development, our illnesses, our health. And I remember seeing a slide. So different conditions have been known for a while to have different amounts of heritability, and people can produce slides showing how, from twin studies, for example, how heritable different conditions are. And there is a spectrum. And basically, it's really hard to find something that kills you or something that gives you ill health that isn't genetic. And I remember someone once saying, well, being struck by lightning's not very genetic. And actually they said, well, no, risk taking, that's got a genetic component. So yeah, I think there will be a genetic component to ME/CFS. I think the slightly more challenging question is, will DecodeME find it? The study has been designed to stand a very good chance of so doing. If it doesn't, that doesn't mean it's a failure, actually. It in itself, that is interesting information. And there have been one or two other conditions where initial results haven't shown, and then people have thought hard and tweaked things and gone back with more people or slightly different categories of people and have got there.
And we will work with anyone around the world to make sure that we have as large a number as possible to find these genetic signals. And that's what's happened in other conditions previously. So thank you, Shona. You've answered another person's question about whether the 25,000 will be enough. Then a question about whether there will be an incentive for pharma to risk R&D, so research and development investment for a new treatment. Well, there isn't anyone from pharma here today, but I would expect and hope that you will join us to go and knock on their doors and insist that they risk such investment. 250,000 people in the UK and growing who have this devastating, appalling disease, all deserve for there to be a lot of investment, and those companies that are listening, and there are several, are very interested in DecodeME and are waiting its results. Thank you for a personal question to me about what led me to research ME. Yes, I have personal links. So I have a friend, a very good friend who has ME, but it's much more than that now. It is something that I wish to learn more about before I reach the end of my scientific career. Will we compare the genetics of ME with any other diseases like endometriosis? Yes, we will, because there is a genetic study, several actually, of endometriosis, so we can do that, and we will do that. So if someone has been diagnosed with ME and long COVID, please do participate, absolutely. Another question was, who can provide the diagnosis of ME? And it's a healthcare professional, I think. So a GP, a consultant, a nurse. I think we, in our frequently answered questions, I think we provide the answer to that there. There's a question about the heterogeneity, the diversity of ME/CFS. And everyone that I've talked to agrees that it is a heterogeneous disorder with very diverse symptoms. That doesn't necessarily mean that it won't have a very narrow set of causes. There are some diseases that affect many parts of the body but have a very specific cause. On the other hand, it could be very heterogeneous because we can split apart ME into these different types, and that's probably likely too. But this is why we're doing a genome-wide study, because we'll be able to see, if we have enough people, enough participants, we'll be able to see that there are these different contributions made. And that might be separable into, for individuals that have a particular symptoms over others. Rather than going narrow and focused, which will be very good for finding causes for those individuals, but not everyone, we'll be able to look at as many people as possible for as many causes as possible that contribute to ME. Will we communicate with all participants with their individual results? We will not be passing back genetic results to participants. And we decided this early after a lot of conversation. And can I ask Shona to summarize why we thought that was a reasonable approach in this case?
Yeah, I fully support that decision. It's also the decision that was taken by the UK Biobank. And we're at a really interesting time in human genetics where there are new initiatives, government-led, NHS-led, really starting to bring genomic medicine at scale. And then separately, there's research initiatives looking to return individual results, how to explain that to the volunteers, how to deliver that within the NHS, how to respect the wishes of the participants, either to know or not to know their genetic results. It's a really complicated area, and I think we felt it would just dilute the main purpose of DecodeME, which is to look for these genetic signals that differ between cases and controls. So there's no return of results, and if that was ever to change, there would have to be a big program of reconsenting and re-explaining. So I think that's really way down the list of priorities. But the genomics world, the human genetics world, is moving fast, and I think in a relatively small number of years there will be a lot more genomic medicine available to people. But before that can happen for something like ME, a huge amount of basic research has to happen first.
Thank you, Shona. A really important question about diversity. Do we in DecodeME have a sufficient diversity of population in the study, e.g. ethnicity? We are inviting anyone who has been diagnosed with ME, who's over the age of 16 years or above, to register. And that doesn't mean to say, of course, that certain parts of our population are less likely or more likely to participate, but we will go out of our way to try and ensure that we are as unbiased as possible in ensuring as many people of all persuasions, all backgrounds, all parts of the UK who can contribute to this study. And the UK Biobank has taken a similar view, and so has sampled from across demographics and across the country, and that allows us to match the controls that we have with respect to their genetic ancestry, etc. But it's a very important point that indeed many people in genetics have paid not as much attention to as they ought to have. There's a question which says that an individual has not received anything in terms of a testing kit or an email or a call. They don't know whether they are part of DecodeME or not, despite, I assume, having registered for the study. So we have not fully launched because we have only opened ourselves up to a small number of people to trial our processes. And when we go to the full launch, then people will be told that they are now invited to log on to the DecodeME website and to answer questions or to request a paper questionnaire. And then everyone will be told whether or not they pass these criteria. So again, we are doing the best we can. It's taking longer than I would have liked, but we are working around the clock to do the best we can for this project for people with ME. Is ME a weakened or overactive immune system? It could be. It could be an autoimmune condition. We've discussed that. We can find out using genetics whether it has genetic overlap with other autoimmune conditions. There was a question before we came online about long COVID and whether we are working with people who are studying long COVID. Yes, we are. We can ask whether long COVID and ME/CFS share genetics links. We will do that analysis, and we are we want to continue to find as many people who are working in long COVID genetics as possible to work with them. There's someone who asked the question about they're concerned that that in DecodeME, they're worried that the DecodeME might use embryonic stem cells. DecodeME does not use embryonic stem cells. And so we hope that your mind is at rest about that. But if you have any worries whatsoever about this, then please go to the DecodeME website and look at the frequently answered questions, because we've covered some of this there. But I also need to say that if, having read everything, you don't want to participate, then you're free to do so, to not participate. That's exactly what should happen, that given all the information that we provide you, you are able to give your consent or not give your consent to participate. So Shona, would you like to carry on?
Yeah, just following on from that, people are also free to withdraw from the study. But actually, you know, we'd almost rather, if you're unsure, ask and try to bottom out the reason that you're not, you know, you're considering not joining, because it's almost better not to join than to join and withdraw. Again, things change, and if something happens and you think, actually, no, I don't want to be part of this anymore, you know, that's absolutely fine. You don't have to give a reason. But I think the key thing is we're doing our best to provide information to help people make the choice. And we're very much there at the end of an email or as Chris says, lots of information on the website. And that will evolve also. We'll be adding to that and have been adding to that, you know, ever since the website launched. So I think that, yeah, ask questions, and it's very much, we want volunteers. There's no coercion here whatsoever.
Thank you, Shona. And to follow up on someone else's question about being recontacted for future research, we ask people whether they don't want that to happen. And so you don't need to opt in to that. You can say, no, I don't want that, but I do want to still be part of DecodeME. and that's absolutely fine. Those people who are happy to be recontacted in the future for future studies, of course, that would be great. And we would make sure that only studies that have proper ethics approval and etc. are ones in which people would then be recontacted for.
Yeah. And that helps us to be agile if new developments arise. You know, people are asking, well, you know, are we going to ask about infection with such and such a virus, or you know, some other comorbidity? And you know, if a good hypothesis comes up, we can create a new questionnaire, and we can go back to the people who've said they don't mind being recontacted, and we could say, look, we think it'd be really useful, really helpful, really interesting if you could answer these new questions. And that way we really start to build a deeper picture and a deeper understanding, and as I say, allows us to be agile and responsive to other research that might be coming out, whether directly from ME/CFS or perhaps from long COVID and ME/CFS post-COVID.
So there have been several questions about Epstein-Barr virus, glandular fever, multiple sclerosis. And this is testimony to everyone's interest in their being able to follow the literature and to read up on the science, because this is indeed one of the major findings of modern times. In January, there was this paper in the journal Science suggesting, with very strong evidence, a link between Epstein-Barr virus infection and multiple sclerosis. Basically saying that individuals who are later diagnosed with multiple sclerosis will have to have been infected with an Epstein-Barr virus. Although the majority of people, of course, do not proceed to multiple sclerosis having been infected. Does this help ME research is the major question. Because as you well know, people with glandular fever have a higher likelihood of going on to being diagnosed with ME than others who have not. Well, we need to do two things. First of all, we need to be buoyed up by the success of the multiple sclerosis findings and the fact that the genetics played a part in those discoveries, and for understanding how the virus predisposes people to multiple sclerosis. And secondly, it might indeed present evidence that there are biological similarities between multiple sclerosis and ME. For example, that they have, they might both be autoimmune diseases, not just MS, but ME as well. And they might have genetic DNA letters that are in common, that contribute to each of those separately. But each of them is a complex disease. So there won't just be one letter that will then be able to be looked at and go, yes, you will get MS, or yes, you will get ME if you are infected with this virus. It is a complex, both are complex diseases, and it will be the rest of the letters, the rest of the genome that will tilt the balance as to whether people will later be diagnosed or not. So there are questions about vaccinations that unfortunately lie outside of my expertise. And I'm sorry, I won't be able to answer those. Then there are questions such as whether some therapies and treatments, perhaps for cancer, might lead to people being excluded. I won't go through all of the exclusionary criteria. In major reason is that it would take a long time to go through them all. And I would just hope that people will answer the questions on the questionnaire and find out whether they are they go through to receiving a saliva collection kit or not. And as I said before, if you're not picked, it doesn't mean to say you don't have ME. It just means that for this particular study, you don't fall into the narrow set of criteria that we have to use. There was a question about, essentially, Shona, are we looking at the whole genome of each sample, or are we just looking at individual parts because of genotyping and using a microarray? Is this something you're wanting to respond to, or shall I?
Yeah, so again, sort of nomenclature. So we are surveying the whole genome. We're looking at points along each chromosome, including the sex chromosomes, although there's not much on the Y. But we're missing the bits in between. So we're just looking at kind of landmarks, fence posts along the way. And as Chris mentioned, we'll have enough DNA prepared from those precious saliva samples that people will be posting. We'll have enough DNA left over after the genotyping, this whole genome survey, if you like, in future to sequence everything with new funding. Sequencing is getting cheaper, and I think the hope would be that if we can demonstrate good success from our GWAS, our genetic analysis of these samples, that there will be a real enthusiasm to find out the rarer variants and the full picture. So I hope that answers the question. We are looking at the whole genome. We're not missing anything out, but we're only seeing a slightly blurry picture. And we hope that in the future we can really focus and have a sharper picture across the whole genome.
Thank you, Shona. And the whole genome includes the mitochondria. We've had some additional questions on that. Yes, we will be looking at the mitochondrial DNA in this first part of the DecodeME study. So there's a question about whether the diagnosis needs to come from a consultant neurologist or from a GP or other healthcare professional. Their concern, as they state, is that many people in the study with ME may not have had access to a specialist to confirm their diagnosis. So there may be diagnoses that are inaccurate, essentially. And it is true that any diagnosis is imperfect in the sense that not everyone diagnosed will actually have that condition. I think the misdiagnosis rate in Parkinson's disease, for example, is about 10%. So we do have to worry about this, and this is why we're taking a lot of time and paying a lot of attention to the questionnaire that we have completely root and branch changed, so that not just do people pass with respect to whether they've been diagnosed or not, but they passed with respect to the criteria, the Canadian or the IOM criteria, but also that the cardinal symptom, for example, post-exertional malaise, that is also evident in people who are registered for the study. So we fully recognize that some people, like with any disorder, may not have been properly or completely diagnosed, and that will change our ability to find genetic signals, but because of the numbers involved, should not stop us from finding those signals. I love this question. Do I have an opinion on whether epigenetics may be relevant to ME? So epigenetics.
That's definitely one for you, Chris.
Oh, I was just about to pass to you, Shona. Bad luck. So epigenetics is everything above the DNA. So for example, there are 4 types of letters in our DNA, and one of them can be modified chemically. It's a methyl group, so that it is different from the DNA that was inherited from your parent. So, and there are other types of definitions of epigenetics, so it can be quite a woolly concept. So to me, epigenetics is all about whether genes are active or not. You have the same DNA in all of your cells, essentially. And the differences in your cells, whether your liver or your toenail or your ear or your brain cell, those differences come from whether your genes are active or not. They're on or they're switched off. To me, that's what epigenetics is. And it's a molecular explanation of those switches. And absolutely, because it's the expression of your DNA, absolutely those switches matter to whether people have ME or any other disease. But this isn't what we're studying. We're studying the genetics itself, not epigenetics. But there's so much data that already exists that is epigenetic data. And so if you have an image that's correct, then have this one, that you have the DNA that we'll get from people with ME and compare with controls. And layered upon that, and layer upon layer upon layer, we will layer with all of the other epigenetic data to allow us to contextualize these genetic signals and allow us to say, we believe that what is going wrong is not just this gene that is in the mitochondria, for example, whose protein is in the mitochondria, but that we think that it's the mitochondria in the immune cell that is going wrong. And we have the tools to do that, and that's what the genetics community at large is doing. And we'll use everything that we have at our disposal to gain insight into what has gone wrong with so many people with ME. So we have reached the hour, and we've ended with epigenetics. And there are still some questions that have come in, and I apologize that we haven't covered them all. To all of those who have reached this point in time, thank you for keeping with us. And I hope that we have answered your questions sufficiently. If not, please do go to the website and see if there's a better explanation there. Thank you to everyone on the team who isn't on this call. The people with ME's contributions have been central and critical. We would not be where we are today without all of those, not just a week or a day, but for the last few years. And thank you, Shona, for all of your insights. Very much enjoyed partnering with you today on this webinar. See you soon. Bye.